Since the dawn of civilization, law enforcement has used many different techniques to prove the guilt of people who break the law. But none of their techniques has changed law enforcement more than genetic fingerprinting. Here are some of the historical techniques that are still used in law enforcement today. Interrogation. Interrogation has been used since the BC era. It is a way to get the truth out of someone by exploiting his or her weaknesses. Police sketches. Police sketches are simply a picture of the suspect taken from the mind of a witness and drawn by a sketch artist. Fingerprints. The identification of fingerprints has been used for over 200 years. Fingerprints are like our own personal signature, right on the tips of our fingers. Blood types. The blood pumping through your veins has a special type called a blood type. Criminologists can use a blood type to narrow down a search between suspects. One of the last historical techniques is a forensic autopsy. A forensic autopsy is when someone searches the exterior and the interior of a corpse to find the cause of death. In 1869, Friedrich Miescher discovered deoxyribonucleic acid, or DNA for short. DNA is the makeup of all living things. It looks like a twisted ladder, and it is unique to every person. For almost 80 years, no one had a way to examine DNA until Rosalind Franklin invented the X-ray diffractor. It bounced X-rays off the DNA molecules and created a picture of returning X-rays. Because of Franklin's discovery of DNA's spiraled shape, her partners James Watson and Francis Crick completed the first 3D replica of DNA's structure. In the 1970s, Frederick Sanger created a way to read DNA through a series of lines and dashes. Scientists and criminologists can get DNA samples from hair, blood, saliva, and other bodily fluids. At the University of Leicester in England, Dr. Lech Jeffrey and his team were studying hereditary diseases in the early 1980s. In the 1980s, scientists were trying to find high variable regions in DNA, not only trying to show that they existed, but to show that they had specific details in them, and that they were also carried down from generation to generation. They began to see that DNA had patterns that differ from person to person. Sir Dr. Lech Jeffreys discovered many different things. He discovered split genes, and he also was the first to discover inherited variations of human DNA. After they switched from studying hereditary diseases to the patterns in DNA, they came up with the RFLP system. The first case that sparked interest in genetic fingerprinting was an immigration case in Ghana about a boy trying to get back to his mother. In 1989, Dr. Lech Jeffrey came up with VNTRs, or variable number tandem repeats, which are long repeating strings of letters of the DNA. Nowadays, when scientists look at DNA, they don't look at the VNTRs, they narrow in on the STRs. STRs are shorter and last longer than the VNTRs do. STR stands for short tandem repeats, and they are spots in our DNA that are almost always different from other people. Scientists still use Frederick Sanger's idea of having the DNA be represented in dashes and lines. After the Ghana case, genetic fingerprinting spread like wildfire. It first appeared in America in 1988 for a sex offender case. As Lawrence Kaltman from the New York Times explains, a new biological test promises to change radically the way criminologists track down murderers, rapists, and muggers. With all the buzz about genetic fingerprinting, there needed to be a way to compare DNA samples faster, so people created the DNA database.
Evolution of a state and national database of DNA profiles has helped a lot too. A lot of crimes have been solved simply by getting some DNA at a scene. They do the profile, they run it through the computers, and they come up with a match.
In 1993, the National DNA Index was created. It is a list of all known criminals and their DNA profile. Criminologists now can match thousands of known criminals against their sample within a matter of seconds. The RFLP system was far from perfect. It took long amounts of time to process the samples, and to make sure the system worked, someone needed to watch it constantly. It was also hard to test DNA mixed with other samples. So, scientists today use the polymerase chain reaction system, or PCR system for short. The PCR system copies the SDRs so that they can have more SDRs with smaller samples of DNA. Genetic fingerprinting has been helpful in numerous other ways than just law enforcement, such as in 9/11 to help identify the victims. Other things that genetic fingerprinting has helped is in the SHOEN project, which helps identify Holocaust victims, reuniting children from the Middle East, keeping track and counting endangered animals, the many migration patterns of animals, even the authenticity of the past Super Bowl footballs. The study of DNA has changed drastically over the years. It has gone from simply trying to read DNA to trying to change the DNA structure of animals. One thing genetic fingerprinting has evolved into is transgenic animals and plants. These are animals and plants that have had their genetic code altered to make them better. Today, the farthest we have come in DNA research is cloning, which is making an exact replica of someone's genetic code. Due to Dr. Lek Jeffrey's work, almost every American with a TV has seen or heard of these TV shows. Forensic Files, Bone, NCIS, Castle, Law and Order, Without a Trace, Red Rum, and Cold Case Files.
What you see on television isn't always necessarily true in real life. And for the purpose of, getting a nice hour of drama, some of the things depicted there just don't happen. It takes longer than an hour, and I think most jurors realize that now. That was a big problem.
The popularity of these shows reflect our fascination with all things forensic, even if we have no idea what it means. Genetic fingerprinting has provided many opportunities to put the guilty away and let the innocent free. There have been numerous people who have been saved by the work of Dr. Lek Jeffrey, and without it, we would have thousands of dangerous criminals still on the streets today. Because of these reasons, the invention of genetic fingerprinting was a turning point in law enforcement and changed the way criminologists catch and stop criminals forever.